Welcome to This Explainer. Today, we're putting on our microscopic detective hats to track down a seriously persistent suspect, the mastitis pathogen, Staphylococcus aureus. If you've ever found yourself wondering why some bacterial infections are so stubbornly difficult to clear up, especially in veterinary medicine, you are in exactly the right place. We're going to break down how this microscopic villain operates and look at the exact clues scientists use to catch it in the act.
Here's our roadmap for the investigation. We'll meet the pathogen, identify the culprit, uncover its pretty incredible immune evasion tactics, and finally, survey the mastitis battleground.
Section 1. Meet the pathogen, the suspect profile. Okay, let's dive right in. What are we actually dealing with here? Staphylococcus aureus is a gram-positive cocci. Now, cocci just means they're spherical, and these guys are tiny, literally about 1 micrometer across. Because they're gram-positive, they're rocking the super thick peptidoglycan layer in their cell wall, which makes them pop as a dark purple when you apply a gram stain. They are non-motile, so no swimming around, and they're facultatively anaerobic, meaning they're perfectly happy surviving with or without oxygen using fermentative metabolism.
But how do these cells actually group together? Well, when they divide, they do it across multiple planes, which creates these irregular, grape-like clusters. It's actually a massive clue for us. The genus name literally comes from the Greek word for bunches of grapes. It's a perfect visual fingerprint to tell our suspect apart from, say, Streptococcus, which divides in a way that forms long chains instead.
Section 2. Identifying the culprit. Forensic identification. So, how do we definitively nail down this specific suspect in the lab? If a cow has mastitis, that utter inflammation we talked about, you grab an aseptic milk sample and run it through a very precise sequence. First, you culture it on blood agar for a day or two at around 37 degrees Celsius. If you spot smooth, opaque, sometimes golden yellow colonies, you hit it with a gram stain to check for those purple grape clusters. But looks aren't everything, right? We need biochemical proof. That means moving on to the Catalase and Coagulase tests to lock down the ID.
Now, what's really interesting here is the Catalase test, because it brings some actual visual drama to the lab bench. The Catalase enzyme breaks hydrogen peroxide down into water and oxygen. So, when a microbiologist drops hydrogen peroxide onto a staph sample, boom, you get this really distinct, visible bubbling as the oxygen escapes. If it bubbles, it's catalase-positive. That immediately rules out Streptococcus, which is catalase-negative, and just sits there without reacting.
All right, so we know it's staph, but which one? Enter the coagulase showdown. The coagulase test separates our major threat, S. aureus, which is coagulase-positive, from the coagulase-negative staphylococci, or CNS, like Staph chromogenes. Quick side note though, negative definitely does not mean harmless. CNS can absolutely still cause intramammary infections and spike somatic cell counts, but the clinical severity just usually isn't as intense as with our main guy, S. aureus. And coagulase testing actually comes in 2 flavors. You've got the rapid slide test looking for bound coagulase, also known as clumping factor, which causes immediate, visible cell clumping. But the gold standard, the definitive one, is the tube test for free coagulase. If that's present, it promotes the conversion of fibrinogen to fibrin, creating a solid fibrin clot right in the tube. And that clot isn't just a neat lab trick, it's a huge red flag exposing the bacterium's pathogenic potential.
Section 3. Mastering Immune Evasion, the Villain's Arsenal.
Okay, let's leave the lab and get down onto the microscopic battlefield. Think about this for a second. How does a tiny 1 micrometer sphere manage to completely outsmart a massive, highly evolved bovine immune system? It all comes down to a really complex arsenal of virulence factors designed for pure subversion. To appreciate the sabotage, we kind of need to look at how things should work. Normally, host antibodies tag a pathogen. That's called opsonization. It's basically a homing beacon so the host's immune cells, the phagocytes, can recognize, engulf, and destroy the invader, which is phagocytosis. Staph aureus survives by completely hijacking this exact process.
Check out this stealth arsenal. First up, it has a physical capsule that literally reduces its recognition by phagocytes. Then you have protein A, which totally scrambles the antibody tagging. And hey, remember that fibrin clot from the lab test? Inside the udder, S. aureus uses that exact same coagulase to build a literal shield of fibrin around itself, hiding from the immune cells. Finally, it unleashes enzymes like hyaluronidase to break down and carve a path right through host connective tissues.
I want to zoom in on protein A for a second, because this is honestly a masterclass in molecular disguise.
Normally, an antibody grabs a bacterial antigen and leaves its tail end, the Fc portion, sticking out like a handle for immune cells to grab. But protein A, it binds that Fc portion of immunoglobulin G backward. It literally grabs the antibody by the handle. By doing this, it completely neutralizes the host's primary tagging system. It is basically a microscopic invisibility cloak.
So the crucial point here is that S. aureus isn't just fighting the immune system, it's actively hiding from it. One of its most frustrating tricks is intracellular survival. It can actually survive inside the host's own mammary cells. By ducking inside, it dodges immune cells and makes a lot of antimicrobial treatments completely useless. That is exactly why it causes these chronic, recurrent infections that are notoriously difficult to eliminate.
Section 4. The Mastitis Battleground. The crime scene. We've seen the weapons, so let's survey the crime scene. Once S. aureus sets up shop, it starts pumping out destructive toxins. The worst offender is alpha-hemolysin. This stuff causes direct cell membrane damage and vascular injury. In severe mastitis cases, it reduces blood flow so drastically that it leads to tissue necrosis and gangrene in the udder. Then you've got beta-hemolysin, causing even more membrane damage and creating that distinct double-zone hemolysis microbiologists look for when culturing this bacteria.
And this brilliantly illustrates how a bug's basic biology dictates how it acts on the farm. Staph aureus is a contagious mastitis pathogen. The infected mammary gland itself is the primary reservoir, and it spreads directly through the teat canal, mostly during the milking process. This is completely different from something like E. coli, which is an environmental pathogen the cow picks up from her surroundings. Knowing this bacteriological difference is absolutely make or break for farm management and hygiene.
And if all that wasn't enough, S. aureus has a pretty wild defense against our medical interventions. A lot of strains produce an enzyme called beta-lactamase. This enzyme literally hydrolyzes. It chews up susceptible beta-lactam antibiotics before they can even harm the bacteria. It's frustrating, but you have to admit it's a brilliant survival strategy. This mechanism leads straight to treatment failure, which is exactly why antimicrobial susceptibility testing is so critical before you just blindly choose a drug.
This quote from the source text perfectly captures the reality of what we're dealing with. Let me read it for you. S. aureus is not simply a bacterium that causes inflammation of the udder. It is a highly adapted bacterial pathogen capable of colonization, immune evasion, persistence, and transmission. It's that exact combination, the sticky capsule, the backward binding protein A, the fibrin shields, that makes it such an absolute powerhouse of a pathogen. Which leaves us with this final thought. We are locked in a relentless, invisible arms race with bacterial evolution. When our microscopic suspect can hide inside our own cells, chew up our antibiotics, and literally build armor out of our own biology, you have to wonder, will we ever fully outsmart it? Thanks for joining me on this explainer, and keep questioning the microscopic world around you.